We're going to discuss the complements. As described in Chapter 1, complement is a complex series of more than 30 soluble and cell-bound proteins that interact in a very specific way to enhance host defense mechanism against foreign cells. Originally recognized in the 1890s as a heat label substance present in normal non-immune serum, complement was so coined by Paul Ehrlich because it complements the action of antibody in destroying microorganisms. Jules Bordet was awarded the Nobel Peace Prize in 1919 for his in elucidating the nature of complement. While complement promotes opsonization and lysis of foreign cells and immune complexes, chronic activation can lead to inflammation and tissue damage. It is the latter that occurs in autoimmune disease. Therefore, complement activation needs to be carefully regulated in order to minimize tissue damage. In addition to the major protein involved, in activation, there are numerous proteins that act as control or regulator of the system. The three pathways for activation of complement will be discussed along the major system controls. Most plasma complement proteins are synthesized in the liver, with the exception of C1 complement, which are mainly produced by intestinal epithelial cells, and factor D, which is made on adipose tissue. Other cells such as monocytes and macrophage are additional sources of early complement components, including C1, C2, C3, and C4. Most of these proteins are inactive precursors or zymogens, which are converted to active enzyme in a very precise order. The characteristics of the main complement protein. So we have here the classical pathway. We have the C1q, which it has a molecular weight of 410, which its concentration is 150 microgram per milliliter. Its function is to bind to Fc region of IgM and IgG. The C1r has a molecular weight of 85, which it has a concentration of 50 microgram per milliliter that it activates the C1s. The C1s also has a molecular weight of 85, which it has also the same concentration as C1r, which is 50 microgram per milliliter. But its function is to cleave C4 and C2. C4 on the other hand has a 205 molecular weight, which it has a concentration between the 300 to 600, which its function is that the part of C3 converts to C4b. C2, on the other hand, has a molecular weight of 102 and it has a concentration of 25 micrograms per milliliter, which its function is to bind to C4b form C3 convertase. The C3, on the other hand, has a 190 molecular weight, which it has a concentration of 1,200 micrograms per milliliter, which its function is to key intermediates in all pathway. The C5, on the other hand, has a 190 molecular weight, which it has a concentration of 80 micrograms per milliliter, which its function is to initiate membrane attack complex. The C6, on the other hand, has a molecular weight of 110 and has a concentration of 45 microgram per milliliter, which its function is to bind to C5b in MAC. The C7, on the other hand, has a molecular weight of 100, which it has the concentration of 90, which the same thing is the function is to bind to C5b/C6 in MAC. The C8, on the other hand, has a molecular weight of 150, which its concentration is 55. The function of it is to start spore formation on membrane. The C9, on the other hand, has a molecular weight of 70, and the concentration will be 60, and its function is polymerize to cause cell lysis. In the protein of the complement system, the serum the factor B has a molecular weight of 93, which the concentration of it is 200. The function of it is to bind to C3b to form C3 convertase. Factor D, on the other hand, has 24 molecular weight, which has only two concentration, which the function of it is to cleave factor B. Properdin, on the other hand, has a molecular weight of 55, which its concentration is 55 to 25. That function is to stabilize C3b to C3 convertase. In the MBL pathway, the MBL has a molecular weight of 200 between 200 and 600, which it has a molecular weight between 0.0002 to 10. Its function is to bind the manus. MASP1 has a molecular weight of 93, which it has a concentration of 1.5 to 12. Its function is unknown. MASP2 has a molecular weight of 76, which its concentration is unknown. The function of it is to cleave C4 and C2. Now, the complement system can be activated in three different ways. The first is the classical pathway, which involves 9 proteins that are triggered by antigen-antibody combination. Pillemers and colleagues later discover an antibody-independent pathway in the 1950s, and this plays a major role as a natural defense. The second pathway is the alternative pathway, was originally called the Properdin system because the protein Properdin was thought to initiate this pathway. Now, however, it is known that Properdin major function is to stabilize a key enzyme complex formed along the pathway. The third pathway is the lectin pathway. Is another antibody-independent means of activating complement proteins. Its major constituents, manus or mannan, binding lectin, adheres to manus found mainly in the cell wall or outing coating of bacteria, viruses, yeast, and protozoa. While each of these pathways will be considered separately, activation seldom involves only one pathway. The complement system plays a major part in the inflammatory response directed against foreign antigen. Although the end product of complement activation is lysis of the invading cell, many other important events take place along the way. Complement fragments acting as opsonin, for which specific receptors are present on phagocytic cells, enhance metabolism and clear immune complexes. In fact, uptake of immune complexes in the spleen appears to be complement dependent. Complement components are also able to increase vascular permeability, which recruit monocytes and neutrophils to the area of antigen concentration and trigger secretion of immunoregulatory molecules that amplifies the immune response. Any deficiency in the complement system can result in an increased susceptibility to infection or in the accumulation of immune complex with possible autoimmune manifestation. Thus, this protein plays a significant role as a host defense mechanism. First, we must discuss the classical pathway. The classical pathway or cascade is the main antibody-directed mechanism for triggering complement activation. However, not all immunoglobulins are able to activate this pathway. Those classes that can include IgM, IgG1, IgG2, and IgG3. IgM is the most efficient because it has multiple binding sites, thus, it takes only one molecule attached to two adjacent antigenic determinants. to initiate the cascade. So IgG molecules must attach to antigen within 30 to 40 nanometer of each other before complement can bind. And it may take at least 1,000 IgG molecules to ensure that two are close enough to initiate such binding. Some epitopes, notably the RH group, are too far apart on the cell for this to occur, so they are unable to fix complement. Within the IgG group, IgG3 is the most effective, followed by IgG1 and IgG2. In addition to antibody, there are few substances that can bind complement directly to initiate the classical cascade. This includes C-reactive protein, severe viruses, mycoplasmas, some protozoas, and certain gram-negative bacteria such as Escherichia coli. However, most infectious agents can directly activate only the alternative pathway. Complement activation can be divided into three main stages, each of which is dependent on the grouping of certain reactants as a unit. The first stage involves C1, which is known as the recognition unit. Once C1 is fixed, the next component activated as C4, C2, and C3, known as collectively as the activation unit. C5 through C9 comprise the membrane attack complex, and it is this last unit the complete lysis of the foreign particles. Each of these is discussed in detail in the following section. Now we're going to do the recognition unit. The first complement component to bind is C1, a molecular complex of 740,000 Daltons. It consists of three subunits which are C1q, C1r, and C1s, which are stabilized by calcium. The complex is made up of one ClQ subunit and two each of the C1r and C1s subunits. Although the C1q unit is the part that binds to antibody molecules, the C1r and C1s subunits generate enzyme activity to begin the cascade. The C1r, the C1s complexes is an S-shaped structure with several domains of unequal size. The sequence of binding is C1s, C1r, C1r, C1s. It is hypothesized that this structure assumes the shape of a distorted figure 8 and it wraps itself around the arm of C1q. C1q has a molecular weight of 410,000 and is composed of 6 strands that form 6 globular heads with a collagen-like tail portion. This structure has been linked to a bouquet with 6 blossoms extending outward. Each of the 6 stalks is composed of 3 homologous polypeptide chains, which are A, B, and C, that forms a triple helix. Alanine residues, which interrupt the triple helix, make a semi-flexible joint that allow the stems to bend. As long as calcium is present in the serum, C1r and C1s remain associated with C1q. C1q recognizes the fragment crystallizable Fc region of 2 adjacent antibody molecules, and at least 2 of the globular heads of ClQ must be bound to initiate the classical pathway. This binding occurs at the CH2 region for IgG and at the CH3 region for IgM. C1r and C1s are both serine protease proenzyme or zymogenes. As binding of C1q occurs, both are converted into active enzymes. Autoactivation of C1r results from conformational changes that take place as C1q is bound. Mechanical stress transmitted from the stems as binding occurs open up the active site on C1r. Once activated, C1r cleaves a Thioester bond on C1s, which activates it. Activated C1RX is extremely specific because its only known substrate is C1S. Likewise, C1 has a limited specific